Hello, and thank you for coming. And thank you to the Department of Neurology for supporting this series of workshops. My name is Luke Hammond, and I'm the Director of Quantitative Imaging here in the Department of Neurology at Ohio State University. I'm really excited to be sharing these workshops on deep learning for image restoration and segmentation because I think they completely change how we can approach image analysis with microscopy data. I've worked with many people over the last few decades on image analysis, both through my time leading the imaging platform at the Queensland Brain Institute and in Australia, and at the cellular imaging platform at Columbia University. I think these advances in deep learning can be applied to many problems that were completely intractable or very challenging just a few years ago. And I'm hoping that by sharing these tools for image restoration and segmentation, it'll allow you to approach projects that were impossible for you to approach or accelerate the research that you're already doing. So I think the most important thing is that's really exciting is we really have the tools now to explore and quantify biology accurately. And this wasn't the case just a few years ago. I think many of the things that deep learning has allowed is really transformative for biomedical research. And so the primary aim of the workshops that I'm running are to equip you with practical skills so that you can use these tools to accelerate and enable your research. And so previously we looked at using pixel classification and machine learning to perform image segmentation. And this pair of workshops are really focused on deep learning for both image restoration, which will be today, and the subsequent workshop on segmentation. And following this, we'll focus on using ImageJ to work with images as well as automate image analysis. So as many of you know, fluorescence microscopy gives us this ability to explore tissue from whole brains to cells, organelles, all the way down to single molecules with fantastic contrast and specificity. And for many of us, we start our research projects with the simple goal of performing some imaging and then performing some image analysis. And we sort of think about the project in this way. Use a microscope, acquire some images, then go to Fiji or Imaris and perform some analysis. And what I'm hoping to convey in this series of workshops is that we need to think about image analysis in a much broader context. So we're not just performing imaging, we're preparing samples, and we should prepare our samples in a way that will best fit getting an image that will be suitable for analysis. And in terms of analysis, we should be thinking in a more sophisticated manner. And so these workshops are helping, hopefully, to let you think about image analysis in terms of a pipeline where we want to restore images when necessary, draw on deep learning techniques to get accurate segmentations of the things we're exploring, and then perform our measurements. And then finally perform statistics and plotting. And in the previous workshop, we talked about things to consider when preparing a sample. So even the fixative that you're using, the way that you're labeling the sample, and the way that you're processing the sample will have a huge impact on the type of images that you can acquire and subsequently the type of analysis you can perform. And it's much easier to solve problems in the sample preparation side than it is to start to troubleshoot at the image analysis side. And so I encourage you to think about these things before you come to your image analysis problem, so that you can have better images to work with. And so many of you might be asking, okay, well, why should we bother using machine learning for image segmentation? I have some ImageJ macros, they work okay. But the real answer is that if you look carefully when you're using these simple threshold techniques, especially when you have complex biological structures, you'll find that it can be hard to find the right set of filters that will give you a consistent result that will work across samples and across conditions in a manner that's robust, so that you could give it a sample from a different condition where the cells look different and still get a result that you could use to compare to, say, your control condition. And so here's an example where we're using just a simple thresholding approach to isolate these cell bodies from the dendrites and axons in the image, and you can see it's a quite difficult problem to solve. Of course, we could do many filters and get a better result for this image, but if we start to apply that to many images, we'll probably find that it fails. And so a good way to get around this is to use pixel classification, and this is where we learn features about the local area of these pixels and start to classify them into different classes, say cell bodies versus dendrites and axons. And the tool that I introduced to you for doing this is Elastic, which I think is a fantastic tool for doing pixel classification. It's very easy to use and has solved a number of problems. So just with some simple pixel painting, we can perform excellent work in EM segmentation, where we're just relying on really structural and feature differences in the image to pull out features of interest. And then we can couple that machine learning technique for pixel classification into classifying objects as well. So we can look inside objects and look at maybe how the staining texture changes as well as the shape of the object and start to classify cells into different conditions. So here we're looking at nuclei that are in different cell states. So how does pixel classification work? So in the case of Elastic, we have an image. So here is on the left is an image of some neurons, just the raw fluorescence colored in a grayscale lookup table. And what we do in Elastic is generate filtered versions of this image. So we generate many different filtered versions, and just to illustrate, I'll show some of these on the right. So just some Gaussian smoothing. So this is really just blurring the image. And so as that kernel size increases, the image becomes more blurred. This isn't potentially that useful, but other filters, such as the one I'm showing here, are useful for highlighting the edges of cells, or other objects in the image, and so this can help define the boundary of cells. And then these later filters are really quite useful for picking up texture or finer information in the images that are more complex than just an edge, for example. And so by using a combination of all of these filters, we can then start to learn, okay, in this pixel location, these are the values that I have for all of these filters. This pixel is more likely to be a cell body, and so on for dendrites or background. And this allows us to get this fantastic image of the dendrites and cell bodies really accurately delineated. The problem with this is these techniques often use CPU. So in the case of Elastic, all of these images are generated using the CPU. And even though it's a very powerful approach, it doesn't really take into context what's happening across the whole image. So we have this sort of local information, but it'd be nice to be able to have some broader context so we could start to say, okay, well, when I see cells here, they're not actually cells, they're actually part of these very dense dendrites that look like cells. And this is where a deep learning approach will work over the sort of Elastic machine learning approach. And so the two problems there are it's slow because it's using a CPU, and we're limited. We're not taking in more information that's really present in the image. So to give you an example of how these techniques can be applied, I wanted to share a collaboration with my long-term collaborator, Grace Shin, who's assistant professor here at Ohio State University, and her talented student, Madison Smith. And so what we're looking at here in green is nociceptive neuron in the Drosophila larva, and we can also see in green immune cells. So these are resident macrophages in Drosophila, and the magenta color is integrin. So this is a protein that's found on the cell surface of some immune cells, but also in the muscle and the skin. And so this is like almost a pan marker that allows us to see many different cell types in the tissue. And the challenge we had is being able to understand what's happening in the sensory neuron as well as these immune cells, even though they're labeled with the same marker. And this is because we wanted to perform live imaging as well as use a number of different markers. And so to solve this problem, we used a 3D UNet that was trained to classify different parts of the immune cells so that we could detect not only the nucleus of the immune cells, but also the cytosol and the membrane. And you can see here just how tightly opposed these immune cells are with the sensory neurons. So this is a very complex image that would be very challenging to do reliably without using these techniques. And now you can see the immune cell mass overlaid. And this is actually an earlier version of the model. We've even improved upon this more recently, but you can see already this accurately delineating the immune cells and even some of the faint cytosol, which is hard to see without close inspection of the image. And so then we can take these large tissue volumes that are required at high magnification and process them very quickly because we're using GPUs for the processing, and then pull out very useful features. So here we can see all the immune cells. So this is just a maximum intensity projection so that we can look at it in 2D, but these are 3D isolated cells. And we're looking at here all these sort of green immune cells, but also other immune cells that are labeled with integrin or the MSN red label, we can pull these out even though we're looking at different colors, and we can do this very accurately. And we can use the same approach to pull out the sensory neurons, so the dendrites as well as the soma, automatically. And this allows us then to do quite sophisticated analysis where we can look at the dendrites and perform analysis where we look at distance from dendrite, distance from soma. We can also look at the local branch density and correlate that with the immune cell density and all the morphological and protein expression levels of the immune cells. And so this allows us to really drill down and see what is changing in these different conditions that we're exploring. And so by having these immune cell masks, we can also extract 3D volumes of every immune cell in the tissue and treat these as kind of a catalog of immune cells that we can then apply machine learning on to classify them at different states of activation. Okay, so how do these deep learning models work? And so I know because there's a varied experience in the background, I want to keep this quite simple, but hopefully in a way that you understand what is happening to use these networks for your own research. And so the way we're solving these problems is by creating an architecture of neurons in varying amounts of layers and in varying numbers to solve problems by learning what is the correct response. And so gradually refining their weights to give us the answer that we're looking for. And so to do this, we have an input and then we have an expected output so that the network can start to learn, okay, when I see this input, whatever it may be, I should be generating this output. And so if we just look at a very simple type of network where we give a series of numbers, you could think of this as maybe some health metrics for a person, like blood pressure or the levels of certain proteins. And then we want to just say, is this person healthy or not? And so we give the network these particular values, and then these are passed through the network, and then it predicts, okay, this person is healthy, or this is the value of 1. But in fact, we know that these set of numbers, the person is not healthy, and so we sort of compare this to the predictor sort of correct response, which is 2, and then we use this information and what's called backpropagation. We pass this back through the network and update the weights of all of the connections between these nodes or neurons, so the network can become better at predicting what to do with this set of input values. And so this process is iterative. We repeat this over and over again until we get closer and closer to what the prediction should be. And that's essentially how they work. And so in the case of deep learning for image restoration and segmentation, we're not just passing numbers, we're passing in image arrays. And so in our case, we have an image of, say, the neurons in the tissue, like the example that I was showing you. We pass this through the network, and then we have an expected output. So in my case, we wanted to see the dendrite branches and the cell soma. But at first, the network is very poor at doing this, and so we get a result. We compare that to our expected ground truth. We backpropagate through the network, update the weights so that the network can perform slightly better at doing this task. Check the output, it should be improved. Compare it to the real output, and then backpropagate again. So iteratively, we get a closer and closer to the result we want. Okay, and so the way this happens is almost all the networks we're using in this workshop are convolutional neural networks. And this can be maybe a little bit confronting if you haven't got a maths background, but I think it's good to know just the basics of what's happening so that you can grasp how these models work and think about them in a way that you can maybe fine-tune the work that you're doing. And so we know from previous workshops that images are made up of pixels, and these pixels are really just sets of numbers, so an array of numbers. So a bright pixel may have a bright value, a dark pixel may have a low value. And the way that we modify these with a convolution is we have our image, which is an array of numbers, and we pass it or multiply it with a kernel. And so the kernel is a much smaller array that we pass over the image and calculate the result. And so this convolutional processing is really the case of an image multiplied by a kernel. And this can be a little bit hard to understand, and so I've just included a slide here to sort of help demonstrate this, and this is just from the NVIDIA's website, but I think it gives a good example. So here we have an array of images, an image that's just ones and zeros, and then we're multiplying it by a kernel. These are the red numbers in the lower right-hand corner of each cell. So the first one is 1 times 1, then plus 1 times 0, plus 1 times 1, and so forth for these 9 cells. And if we add those all together, we get a value of 4. So from this image, we're generating a new image. And you can see that this image is a little bit smaller. That's because we can only pass this array across the image 3 times for one row. And then ultimately, so if we go to the next cell, we do the multiplication for this. So 1 times 1, 1 times 0, 0 times 1, and so forth. Add these all up, we get 3. And so as we go along, we'll fill up a new image array. That's the combination of this kernel of ones and zeros multiplied by the green array. And so this is our new image that has the convolutional feature of that kernel. And the way that you can think about this in real images, so here's an image that's just a grayscale image. It's a little bit blurry. What I can do is apply a sharpening filter. So you can see a sharpening filter has a very sort of high value in the center, and then negative values or 0 values on the sides. And so as we pass this kernel across the image, it becomes sharper, or we see more fine details. We have Sobel filters, and so these are oriented in a particular way. So you can see this left Sobel has higher values on the left-hand side, negative values on the right-hand side, and zeros in the middle column. And if we apply this to the image, then we can start to see vertical lines are starting to stand out in the image. And correspondingly, if we use a top Sobel filter or a kernel, then if we pass this across the image, we can start to see horizontal stripes in the image. And so there's many different combinations of numbers that you can use in these kernels. Here's an embossing kernel that helps features stand out in a more embossed style. And when we're using these kernels in neural networks, these are the sort of values that we're optimizing in the kernel to start to pull out features in the image that are useful for determining where the mask should go. So some combination of these filters, in fact many of them, will help to determine in this pixel, that's where the neuron cell body should be. And importantly, this actually happens not only at a low sort of local area, like in the pixel classification, but it's also happening at a global level for the whole image. And this allows us to learn contextual information for the whole image as well as this more local information. And this is really why these deep learning approaches work so well. So to give you a bit more of an understanding, the convolutional neural networks that we use specifically for these image segmentation and many restoration problems, what's called a UNet. And UNets are known for this because they have this encoder phase where the image gets smaller through various filtering, which I'll explain, and then gradually becomes bigger during the decoding phase. And so this sort of style, which has a U shape, is the reason they're called UNets. And essentially, we're taking an image. Oftentimes we have an image, we'll need to tile it. So we're taking here a tile of an image that has a specific size. And then we perform, you can sort of see in the bottom right, there's a legend for this, but we have a convolutional filters, like I explained to you. So these are 3x3 filters, just like we were looking at before. And there's many of these applied. So from one image, we end up with 64 images because we apply 64 of these convolutional filters. And then as a result, the image gets a little bit smaller. You can see the dimensions changing here. And then we do what's called a max pooling. So this is where we basically pull an array of 2x2 pixels into one. So this sort of has the effect of downsampling the image as we go. So as you follow the encoder, the image gets smaller and we get many more images because we're applying many more of these convolutional filters. So the image gets smaller and smaller and has more of these filters. And so you can, down the bottom here, you can see there's hundreds now of these images that have been filtered in different ways. And then as we go back up on the decoder side, we're upscaling the image. So we're using filters that help to restore the resolution of the image, and we're also pairing that with the information from the encoder phase. And so really the information is connected together, and this allows us to really pull out features in a very accurate way of where the boundary of a cell should be or where a dendrite should be, including the context of the whole image. And so we can work our way back up, and then ultimately we get these excellent segmentation results that just can't be achieved by any more conventional approach. And of course, the applications for this should be obvious. Image segmentation, which is a big problem for many of us, can be really well solved using the UNet approach. Image restoration, which is an incredibly important technique for being able to image quickly or image in live samples. And the combination of these two really allows us to push the boundaries of what's possible with microscopy. So there used to be, and there still is, this trade-off between image resolution and signal and speed, but we can really stretch those boundaries. So now we can image a lot faster without compromising our ability to see cells or see endosomes. And I think this is really a watershed moment in what's possible using microscopy. And I hope that these techniques that you learn during these workshops can really help you think outside what you thought was previously impossible and do exciting research. And so in the case of image restoration, we can take these extremely noisy images that would just be impossible to work with in the past, or exceedingly difficult to work with. And by having a network that's trained to understand, okay, when I see this very noisy image of some nuclei, this is actually what the nuclei should look like. And so we can use this network then to give it new images of these nuclei at very low, high-speed imaging, very low signal, but restore them into a useful image. When we're talking about image restoration, I think it's useful to actually understand a metric that we use for comparing images. And so you often hear people talking about signal to noise, and you may not necessarily understand what that means. And so I thought it would be good just to explain that. So here I'm showing you two images. One is obviously better than the other, and you might just say, okay, one is good, one is bad, but how do we define that in a way that's meaningful? And so we would use signal to noise to really quantify the difference between these images. And the way that you can perform this calculation is by looking at the signal. So this will be like, say, the signal in the cell body or the dendrite, whatever you're sort of trying to look at specifically, minus the background. So this should be an area in the image where there's no features. So this can be a small area where there's just really no signal at all. So you should be careful about where you place this area for measuring. And then the standard deviation of that measurement in the background. And this will allow you to report the signal to noise. So you can see on the left, our signal to noise is 94. On the right, it's 6.78. And so this is really a good metric of saying, okay, well, this is how different these images are. And this is also a metric you can use to sort of report how the image is improved by restoration. And to give you an example of how restoration is used I think a fantastic example is in live imaging, where we're often constrained by the signal that we can detect and also the sensitivity of these samples to the light that we're shining on them. A lot of times if we're looking at protein trafficking, if we shine the laser bright enough to see what's happening, one will bleach the protein, and so that's what's been shown here in the top row. But also we could perturb the biological processes. We might be looking at something related to cell division or protein trafficking, and when the cell starts to be affected by this high laser power, the dynamics change completely. And so what we're reporting has no relevance in biology. And so the alternative is we image with very low laser power, but then we can't see the endosomes at all. And so this is kind of a difficult situation to be in. But by using image restoration, and in this case, Harry Schroff's lab is using a technique they developed themselves called RCAN. They're able to image with the low settings that you're seeing in the middle row, but restore to images seen in the bottom row. So then we can image quickly, but still have images useful for analysis. And in this case, we're looking at mitochondria and lysosomes, and by using this technique, they could look at interactions between these endosomes in live cells at a speed where they could detect meaningful sort of changes in dynamics without perturbing these biological dynamic events by using low laser power. And myself and colleagues have actually applied this in the case of neuron analysis. Many times when we're imaging neurons, the images we get back are not ideal. They may be imaged in tissue. There could be scattering that's affecting the quality of the spines. We also have anisotropy. The axial resolution is much worse than the lateral resolution. And so by using a series of restoration techniques, we can go from the image on the left to the image in the middle, where we have close to isotropic resolution of the spines on the dendrite. And then we can use a UNet to pull out the spine heads, the neck, and the dendrite to allow us to do automatic analysis of spines in a way that is much more accurate and reliable than previous approaches. And so this paper is actually up on bioRxiv now. The technique is called RESPAN, or Restoration Enhanced Spine and Neuron Analysis. And this allows us, as I was showing you, to get very nice 3D renderings of the neuron. Okay, and so a common technique and one that we'll be looking at today is CARE. This is one of the early papers really demonstrating how well this image restoration could work. And they shared the code in a way that's actually very accessible for people to start applying it to their own work. And so CARE stands for Content-Aware Restoration, and it's really a case where we're training a network to understand, okay, when I see this image, the image that I should be generating looks like this. So we're pairing images of low signal to noise and high signal to noise. And these are just showing examples. I think it's just amazing how low the signal can be in the input images and what you can get back in terms of structure and contrast. And these techniques have also been applied to deal with this issue of anisotropy, or missing slices in Z. So you can see in the top panels here, we have a large Z step, and so it's hard to really resolve the 3D structure of the nuclei labeled in magenta, and even the membrane around them. But once we use this technique, we can actually start to see the 3D structure of these cells much better. And you can really think outside the box in how to apply these techniques. I think this is one of my favorite examples. This is from the RCAM paper from Harry Schroth's lab, but I think it's just a fantastic example of how far you can push these methods. In this case, instead of just trying to use restoration approaches to restore images at the same resolution, this approach tried to restore images to an expanded version of the sample. And so the way that they applied this technique is they took an expanded sample, so using expansion microscopy to expand a cell sample to be much larger than it really is, imaged that using their microscope, then convolved or processed that image, basically blurred that image to be similar to how it would look if it wasn't expanded. And use that as one input to the model. And then as the other input, they use the deconvolved version of this expanded image. So on one side, they have this low resolution blurred image that they would normally see on their microscope. And they're training the network to do its best job as possible to make that image similar to the deconvolved high contrast image from the expanded sample. And so you can see on the right how this process works. So they basically have two models, a sort of denoising model, like we discussed, and then this expansion model to restore the resolution. And I think that the images sort of speak for themselves in what they're able to resolve from this low resolution image. So basically they can image the samples normally on the microscope and then restore to something that's similar to if it was expanded. Of course, they can't go the full way, but the improvement is actually quite impressive. So 1.9 times laterally and 3.6 times axially. So this is, if you're trying to study the morphology of these endosomes, this is an incredibly powerful approach. And similarly, SelfNet is an approach to help deal with the same issues with anisotropy in images. So we often use wide field or confocal techniques where the axial resolution is poorer than the lateral resolution. And these techniques can use the images that you acquire to infer what the axial resolution or axial image should really look like. So by learning features laterally, blurring them the same way as they would appear if they were present axially, you can start to restore axial images to be closer to lateral images. And you can see the effect of this type of restoration here in the image. And so this is, I think, very powerful for anyone who's interested in morphology and structure and trying to get closer to what those features should be in their real size. And again, just demonstrating how this works in 3D. And lastly, I wanted to touch on the models we're looking at today. These are all trained models. So we have these pairs of images, but there's a lot of work going on into untrained models that can just know how to denoise images. So these are models that you can get. You don't have to do any more training, but you can apply them to your images and get good restoration results. So we won't look at that in this workshop, but it's good to be aware of them. And this is a new one, UniFMR, which can actually perform quite favorably even to CARE. So you can see here, this is the results. At the bottom here, they're looking at this NRMSE. So this is basically the mean squared error. So this is really just the difference between images. And so lower values here really say that, okay, this image is actually very close to the ground truth image. And they're showing is really this method can get very close to the ground truth compared to other methods. Okay, and lastly, I think because it's important to understand how to create these training sets, so we're looking at how to use these networks, but later on you'll want to do this for your own data. And so the example data that we'll be working in the workshop on, this has been acquired using low signal to noise and high signal to noise parameters. And so for low signal to noise, you can see here I was using 10 millisecond exposure times, no averaging, extremely low laser power. So if you were using this for your experiment, you could image very quickly. So if you're doing volumetric imaging, that means you can image large volumes in a very short period of time. If you're doing live imaging, it means you can image very gently. And then for the high signal to noise image, these are my ideal images, what I'd like to actually have if I was at the microscope, but maybe I don't have time for, or I can't treat my sample with that much laser power and expect it to survive or bleach. So in this case, I use 300 milliseconds, but then I averaged 8 times. So these are really long acquisitions, but give me very good contrast and really allow me to resolve the features. And then what I can do on top of that is deconvolve the image. So what I'm doing is comparing low signal to noise images to high signal to noise images that have been further processed with deconvolution to restore resolution and contrast. And so that's how you should think about acquiring these types of data sets. And importantly, these images need to be exactly aligned. So the best way to acquire them is really one after the other as quickly as possible. So if you'll need many of these images, at least 10 volumes is a good starting point. Don't go and acquire all the low signal to noise images and then come back and acquire all the high signal to noise images, because the stage may not, maybe there's been some drift, or maybe you won't get back to exactly the same spot. And so it's very important the pixels are perfectly aligned, and so I would acquire the low signal to noise image straight after the high signal to noise image, so they're perfectly matched. And finally, I just want to touch on a word of caution. So whenever we're applying these deep learning techniques to our images, we're training them with paired images. And so the network, they're very powerful because they have a lot of information about what's happening in the image. So they perform much better than just deconvolution because they understand what's happening in the image, they understand the noise, they understand the features that are in the image. But because of that, they also have a limitation. And they've learned features in an image. And so if you try and, in this case, we're looking at actin filaments. If we use a network that's been trained to detect actin filaments, it will work quite well. But if we use a network that's been trained to, in this case, look at DNA images, you can see it's actually starting to lose features that are there in the image. And so when you apply a network that's been trained on a set of images with a specific set of features or a specific magnification, and you use that on a new set of images that don't match those parameters, you can expect it to hallucinate features that don't exist, or start to hallucinate away features that are of interest, or just perform very poorly. So always train these networks for a specific purpose and use them just for that purpose, and keep that in mind for both the restoration and for the segmentation. And with that, I'd like to move on to how we can actually use these image restoration techniques in Google Colab. Okay, thank you for your attention. Okay, so for this portion, we're learning about how to use Python for image restoration. So I think to start off, I know some of you don't have any experience at all using Python, and so I've shared these 3 Google Colab notebooks with you, basically starting from the very early stages of using Python just to get a grip with how to code, and then we'll move into how to use a restoration model, and then we'll finally train a new model using our own data. And so along the way, I'll just really go through this for someone who has no experience with Python. So for those of you who do have experience, bear with me, and we'll get to the more interesting parts soon. In terms of image analysis, I think Python is an essential tool alongside ImageJ. I think ImageJ, or Fiji, is fantastic for viewing images, for doing small tasks. And it's great because you get a visual feedback on everything that you're doing. But once you refine what you intend to do, whether it's processing a large number of images or using these deep learning tools, Python is a much better solution because it's more efficient. It allows you to use state-of-the-art deep learning tools very easily. There are ways to do it in Fiji as well, which we'll cover in later workshops, but it's very efficient to use these new tools within Python, and it allows you to do things in a high throughput manner. And so you can tie it in with, you have your big data, you can use these deep learning tools, you can use other segmentation and analysis tools, and then you can have your plotting and statistics all together in one processing script. And it's difficult at first, I think, for people because you're just working with text and you don't have this visual feedback, but there are ways to get visual feedback, which I'll show you. But really these tools work together. So think of Fiji as a way of interacting with your data, exploring results, or looking at data before it's used in Python. And then think of Python as a way of using these state-of-the-art tools and creating high throughput scripts, or even just knowing how to use other people's scripts to process data efficiently. So when you start to use Python, it can be quite confronting because you don't have this interface. You just have to type in commands to get the result that you want. And starting with a blank canvas is always very challenging. And maybe when you look at some code, you don't understand what's happening in Python code, and so you feel too intimidated to start using someone else's scripts as well. And so just to start with, I want to share the very basics of using Python. And so we're using Google Colab for this workshop because it's something that will be consistent for all of you. And so when you're using other people's code, one of the challenges will be your Python environment may be different to theirs. And so with Google Colab, we can be assured that all of us are using the same environment. That means it's the same version of Python. It has the same versions of packages that we might want to use. And then we can all install additional packages if we need to, and they'll be the same version too. Later on, especially if you have a more powerful workstation, it can be better to use Python locally. And in that case, you might use Anaconda to create your own environment for a particular analysis program. And this is somewhere where you can get stuck, but if it's a good program, they'll tell you exactly what to install to make it work well. So once you have an environment, either in Google Colab or locally, the first thing that you'll need to start doing is importing the packages or tools that you need to use to run the script. And the best way to think about this is, if we use the analogy of an artist, if they're about to start work, they'll need to collect tools that they'll use to create the work. So in Python, the way that we import those tools is by saying import, and then we might name the tool that we need. So if we're an artist, we might say import paints. That way we can have access to all of our paints. We can be more specific when we're doing this. We can say from paints, import blue. And that way we're just specifying, okay, we just want the blue color. We don't need all the paints. We just need blue. And the other thing you might see is naming the package is something more convenient. So maybe we can say import paints as P. And that way, instead of having to refer constantly to paints, we can just say P blue. And that way it knows to get blue from the paints. So this is just one way to think about packages. And once you've done this, here I'm just typing and then pressing enter to create a new line. I'm using Google Colab Pro. So this is just, that's where you can see it auto-populating. And I think if you're new to coding, I think this is worth getting because it will help you learn the right things to write so you can start to memorize some of these simple commands. But this can be very helpful. So, okay, I just wanted to talk about that first because that can give some context to the things that we'll be doing in these additional collabs. Okay, so the very first collab that I shared with you, and this is just going from knowing nothing about Python to just getting a general grasp of Python in the context of image analysis. This may be fast for some of you, or maybe it's very slow for others, but I think just to get a basic foundation of what's happening when we're working in Python, I've made this collab just to explain those very simple things and hopefully give you some tricks to work with images. So here, the very first cell that we're looking at variables. So these A, B, C, D, this is what we call variables, and then we're assigning them a value. So the thing to understand with Python is there's different types of variables. So in the case here, we have a 1, so this is an integer, it's a whole number. We have things called floats. These are values that have decimal places. So like 1.5 would be a float value, not an integer value. If it's in quotation marks or if it's any text, then we call that a string. And then finally we have true or false or Boolean variables. And so when we assign these values, what we can do is sort of ask, if we're having some trouble, maybe we're getting errors. Oftentimes you'll get things called type errors, and it's because maybe you're trying to do a calculation with a string. And so if you look and see, okay, I'm trying to add A plus C and I'm getting a type error, it's because C is actually a string, it's not an integer. And so then you can convert that string to a number or vice versa. And so here, just to explore this data, it is helpful here with Google Colab because it's already auto-populating for us. We can fill this in and then just run the cell by pressing Shift-Enter. And so what we have is now we're printing these variables, so it's just basically printing them out here. We can see they all kind of look the same when they're printed, but when we look here at the type, so putting type and then the variable will actually print the type associated with that variable. So you can see here int is integer, float is floating, as I was describing, str is string, and Boolean means is true or false. So that's a very basic look at variables. Now when we start to work with scripts, it's good to have some feedback on what's happening. And so, I'm sure if you've done any coding, you'll see these examples like print, and then we can say, hello or something like that. And if we run this cell, then what we'll get is, it'll print hello to us. But when we're coding, especially when we're doing image analysis, it's good to have some feedback about what's happening in our analysis so we can maybe find where there's an error or see that something's not working right. And so, for example, we might have a number of files or a number of folders. So let's just say we have 10 files and 2 folders. This would be something that our script will be automatically counting these, but for our example here, I'm just creating these variables from scratch. And then we want to print those out. And a good way of doing this, and I've included it at the top there as a reminder for you, is to put F before you start typing in your string. So, and again, Google Colab's filling this in for us, but essentially we would say something like there are, and you use curly brackets to place a variable inside the string. So here we could say files and then curly bracket, and then we can go on with the string. So this way we don't have to sort of do anything fancy. Just by putting F here and then putting our variables in curly brackets, we can have a very simple string line that will just tell us, okay, there's 10 files in 2 folders. So just keep this in mind as you're working on scripts, or if you have a script and you just want some extra feedback on what's happening, using this F followed by the string and the curly brackets can really make a very quick way of printing what's happening. Okay, so we're getting on to numbers. So when we're working with images, when we import them into Python, they become arrays. And so an array is just the same as when we're looking at pixels in the image. If we're looking at this here as a 3x3 array, you can imagine it as an image that has 3 pixels at the top, 3 in the middle, and 3 at the bottom. And so if we just, so NumPy is the Python tool for working with arrays. And so here what we're doing is we're just creating an array from scratch. So we're just basically saying, okay, make an array that has these numbers at the top, these in the middle, and these at the bottom. And then we can just say, let's just print this array. So we'll get to see what it looks like. And this can be a little bit confusing, but we're just really like this top bracket here and the bottom bracket here is the whole array, and then we have each row in its own set of square brackets. And if you're working in images and you never work with code, then this can be just not easy to look at, and you shouldn't have to look at it that way necessarily anyway. So what I wanted to show you is just these arrays can be very easily looked at as if they are images. And so if we import matplotlib, which is a way of plotting images and graphs inside Python, we can then say, okay, I want to plot my array. So this is the array we created here, using a lookup table. So Veritas is a lookup table that's very useful for looking at intensities. It's a multicolored lookup table. We also can plot a color bar, so people know when they see that particular color, it's this particular value. And we can make a title, and then this plot show just will create a visualization of that array. So if I hit enter, now you can see this array that was difficult to look at before is now a visual array. And so you can see here, this value 5 has been given this color here. And so we're basically creating images out of these arrays. Okay, so that's just to understand how images are represented. We can now start looking at how to look at actual images inside Python. And so whenever we're working with our data on Python, we can connect our Google Drive so that we can put data in there and then process it using Google Colab. And so you'll often see these two lines here to be able to connect your Google Drive. And so we can just play this. And then what will happen is it will ask you, okay, is it okay to connect your Google Drive? And then off screen, I'm just accepting that that's okay. Okay, so now I've mounted this drive. Let's actually start to look at images. And so to look at any of the TIFF files that we have, and these are all in the folder that I shared with you for the workshop, what we can do is import TIFF files. So TIFF file is a way of importing TIFF images, and many of you will be working with TIFF images for your analysis. And I just like to actually, the main two things we're doing is like reading and writing. So when I use TIFF file, I generally do from, and then comma, and write. So we're basically just importing these two tools from TIFF file. And then what we can do is we can import one of these TIFFs as an array. So we can just call, instead of just calling it an array, let's just call it an image. And then we can say imread. So we're using the imread tool that we just imported. And here we can give it a path to our image. And to do this, it's actually very easy. So on the left-hand side here, we can access our Google Drive. And if we go in here under My Drive, you'll see all the data that we have on Google Drive. And so I just want to go into where we have the workshop data stored. Yeah, so here's our path. We can copy the path. Let me just close this. And then we can paste that in here. So let's just paste that path. Okay, so it didn't pass with some quotation, so we should actually just make sure either we use single or double quotation here. Go back to the beginning here and place the other one. So we're giving this as a string, and it won't be very useful, but let's just say like print image, and then you'll see again we'll have an array that'll just be many numbers. But that actually represents our image. Actually, what's also useful is we can find out a little bit about this array. Maybe we want to know, is it 2D, is it 3D, how big is it? So that a useful command is image.shape. So this is where we're taking the image and we're asking what the shape is. So now you can see, and so now by printing the shape, we can see, actually this is a 3D image because you can see that there's 3 axes here. And importantly, this is something that, especially if you're working with ImageJ, Python works with the axes in opposite order. So in fact, this image is 465 pixels across in X, 459 in Y, and then 30 slices deep. So this is Z, Y, X. So something to be aware of, especially if you're coming from the world of ImageJ, where we're usually thinking in X, Y, Z. Okay, so let's actually look at this image. So what we want to do is look at the image, plot the color bar, and then see what we get. So when we do this, then this is, I'm going through these things because as someone, if you haven't used Python before and you start to try and do these things, you'll get these kind of errors and it can be very frustrating and you may just give up. And I wanted to show you that you're very close even when you're doing this. And you just need to think about things slightly differently. And even though these errors can look very confusing, if you have a little bit of patience, you'll find that they actually can be quite informative. And so here I'm just trying to plot the image like we did before. But the problem is this is going to create a 2D image, but the image that we have is 3D. And you can see right down the bottom here, we're getting a type error. It's saying, okay, we have a shape that's 30 by 459 by 465, but you're asking to plot a 2D image. It's just not possible. And so here I wanted to introduce slicing. And so slicing is where we have an image, but we only want to look at a portion of it. And the way that you can slice an image is by using square brackets. And we know from our shape before that we have this particular shape in the image. So the very first one is our Z slices. So let's look at the middle slice. So if I say 15, then if we want to look at the full 2D image, we can just put a colon here, followed by another colon, followed by another colon. And so essentially we're saying, I want to look at the 15th plane in the image, and I want to look at the whole 2D image. So let's just run this again. And now we can actually see the image. The same thing works for here. Like, we don't have to look at the whole image. Say we're only interested in that the bottom left corner, for example. Then what we can do is say, only show me the values. And so if we wanted to look at the bottom left, you can see when they're numbered here, 0 to 400, and then 0 to 400 here. This is y-axis, this is the z-axis. So first of all, for the y-axis, we want the high value. So let's say 200 to 400. And then for the x-axis, we want the low value. So let's say 0 to 200. And if we run this now, we'll get a much smaller crop version that's just focused just on that area. So we're not changing the data here, we're just isolating an area of the image to look at. And so I just wanted to show you this because this way you're using Python in a way where you're getting visual feedback on the images that you're working with. Okay. The next thing I wanted to show you here is how to plot image channels. So let's just import another image. So I'm just going to go import image again. So image equals imread, and let's just give a new path. So this will be another example dataset that I shared with you. That will be some cells dividing. So let's just close that. And we're going to paste in here the path. All right, so let's just try this and see what we get. So it's reading in the image. Actually, it's worked straight away. So I've given you the correct code here. So we have, let me just scroll down so you can see clearly. So what we're doing here is we're reading in the image. We have our figure and axes. So this is basically just creating a plot. I don't want to go into too much detail here because I want to get onto the more interesting things, but just to give you an understanding of what's happening here, we're creating the image, we're isolating the image, and then specifically we're looking at a particular slice, so the third slice in this 3D volume. We can actually, let's just so it's very clear. So before we create this figure, let's just print the shape so we can all look at that together as well. So, okay, so we've imported the image, we're printing the shape. Here we have Z slices, channels, and then Y and then X. And so what we're doing in our slicing here is we're saying pick slice 3, and then I equals the channel, and then we're doing the whole 2D image, so all of Y and all of X. And so then what we're saying is, we're making a 2-panel figure, and so basically we're saying for panel I plus 1, so we always start with 0 in Python, so channel 1 is actually channel 0, and channel 2 is channel 1. And then we're saying, okay, so this is channel 1, and then for the next one we'll plot channel 2. And so that's how we can easily get an image that is showing us 2 channels at once in Python. The good thing about Python is, once you have this code, you can just copy and paste it and then change a couple of values. And so even though it may seem a little bit slow at first, you can build up a set of tools that will allow you to explore and work with data very quickly. Okay, so let's just move on. I don't think necessarily Python is the best tool for exploring data in a live way, but when we're working in Google Colab or in some of these Jupyter notebooks, you can kind of create an interactive workbook. And the way we do that is by using these IPy widgets. And so this is just an example here where we can take our image and we're using the Z slice as something that we want to interact with. And so essentially we're saying that for our image slicer, we can use the sliding tool to change the Z position. So if I run this cell, now you can see in the bottom here we have a slider where we can adjust the Z slice. We're just looking at channel you can see here we're just looking at channel 2, which even though it says 1, it's channel 2. If I change this to 0 and we run this again, then we'll be looking at channel 1. So just to show you, it is possible to do some interaction, but I myself would probably prefer just to export this image and then explore it in ImageJ. But if you need to do it inside Python, you can use it like this. Okay. Lastly, it's very useful when we're working with these 3D datasets to create a maximum intensity projection. This is something you might do in other software like Fiji or other commercial software. This is just a very simple command in Python. And so when you're creating image analysis pipelines and you want a quick visual feedback of a maximum intensity projection, all you have to do is use this numpy.max tool, and then you can give it the image. So let's say we can use the cell image that we already have. And here it's saying axis equals 0. So the axis is the one that we want to flatten. So in our case, z is the very first axis. So z axis equals 0 is actually correct in this case. So if we do that, then we can run this. And now we have a flattened image. Actually, let's just run we can do print image.shape, and then let's do print mip.shape, just so you can see the shape of these arrays after we run this. And so you can see, okay, first we had this 3D image, now we have a 2D image with both channels preserved. And if we want to look at that, then we can just run this here. Okay. If we want to look at that, then we can just come down here and say, okay, let's just show the first channel, and we'll use greens as lookup table. And if we click play here, you can see we can see the green channel. If we want to look at this in a different way, we can do Viridis again. Okay, and this is a good example of, okay, so this didn't work. You can see here, these are the different lookup tables that are available. And so if you ever get an error, like I encourage you just to, before you give up, just have a look and see what's actually happening, and you'll find that you probably can actually interpret what's going wrong. So the lookup table that I'm thinking of, well, let's say just use the colorblind version of Viridis to, I think in our case Viridis is actually lowercase v, but we can try that with Viridis. Yeah, and then the colorblind version, if you're trying to create plots that are easy to interpret for people who are actually colorblind, then you can use this version. Oops. Yeah, and so again, we're using a different color lookup table. And if we want to look at the carefully, just to show you, again, like before, we know this code now, so we can copy a line from this code and put it down here in this code, and very quickly you can just start to build up useful images, building on what you already know. Okay, let's move on. So next, what I wanted to show you was how we can actually save these images that we're creating. So in our case, we have this imWrite feature as well, we can use to write out our images. We might have the path where we have a folder that's full of images that we want to create MIPS for, just like the image we made earlier. And so what we can do is we can tell Python, okay, this is the folder where all of my images are saved, and these are all ZStack images. I have an output place that I'd like to put them, so that this is really useful. In Python, we can just say, rather than having to write out a full directory again of where to save these images, we can just say our output path is the input path plus a subdirectory called MIPS. And then, maybe this directory doesn't exist. So a very important line here for us is that this output path, what we want to do is make that directory if it doesn't exist. And if it does exist, just go ahead. So either way, we'll proceed. And then we want to create again, we're just processing one image. I'm going to show you how to process multiple shortly. But basically, we would say, okay, the output image path would be this output path plus the file name. So whatever the image is, in our case it's the MIP image that we created before, we would just say, okay, the MIP path, this is our output path, let's just put this in here. We would just say, write out the MIP image, the path, so this is where the image will go. MIP is the image that we're saving, that's the one that we made just a few moments ago. And these are important to use with ImWrite as well. So I always use image j equals true and photometrics min is black. It just ensures that when you create this TIFF image, when you open it in image j, it's going to look as you expect. So it'll have a, 0 will be black, it will look as you expect. You don't need to include this bottom line here, so you could just close this off and just say, ImWrite, the path, the image, and then this extra arguments. But if you know the resolution of the image, maybe you know the step size and the scaling, then you can include the additional metadata here. So you can say, okay, well, I know that the resolution in this image was 1.5 microns per pixel, and so I can actually put that here, and then when I open it in ImageJ, that's preserved, and that can be useful if you plan to do measurements in ImageJ afterwards. Okay, so we can just run this cell, and then we know now that the image has been created. Okay, so the whole purpose of using Python, in addition to doing deep learning, is so that we can do things in a high throughput manner. So we want to be able to do things in a loop. So we want to iterate over images and process things without having to do things manually step by step. And so the way that we do this with Python coding is by creating loops. And so this is just a very simple example that I wanted to show. So we have a start point and an end point. This could be files in a folder. And so here we're just looping through that series of numbers. So for the number in the range, starting and ending, so start of 1, end, this is the last value plus 1, do X. And so in our case, we can just print the number, but we could very easily change print number to save ImWrite and save the image as a TIFF. And so I just wanted to show you, this is the sort of way of writing a loop in this case that would process files in a very simple way, so it's clear as a starting point. All right, so let's move on now and actually process images in a folder. It's the sort of thing that we would be trying to do in microscopy. So we have our import OS, we import NumPy so that we can work with the arrays. And then we have our TIFF file where we're importing in read and write. So this is what we would need at the top of our script if we were going to batch process images. We need to have an input path. So this is where our images exist. I'm just going to go up and copy this from our path before. And so here I'll put our input path. All right. And then we want to have our our output path. Okay, so then again, this is basically just another way of dealing with creating folders. We're just saying, okay, if the output folder doesn't exist, let's make it. And then we want to loop over all the files. So here we're saying for file name in the directory that we listed here, if that file name ends with .tiff, this is really useful if the folder contains CSV files or also contains JPEGs or other data that you don't want to process using the script. This script is basically going to go through, find all the files, and then only process the files if they end in .tiff. And then we want to make the path to that file. So we say, okay, add that file name onto the end of the input path. So we have a full string that would be this path plus the file name. And so because we have that, we can then use our TIFF file imread to read in that image. So we're making a new array then called image stack. And so if we then, just like we did before, we could just print the shape of that, and we would be able to see, okay, this image has the shape that just like we were looking at before. Then we want to create a MIP of that image, and we're using the 0 axis. So it's basically going to flatten the image along the Z axis to create 2D MIP files, something that you would do very commonly and may even do manually at the moment. We can do this very quickly, automatically, just using these few lines of code. And so finally, after we create that MIP, we want to create the output path. So we're saying, okay, join the output path, which is actually our input path plus MIPS, plus the file name, and we want to differentiate that file name from the original file name, so we're just saying put mip at the front. So it'll be mip plus image 1, mip plus image 2. So that can be useful, especially if you're saving to the same folder, you don't want to get these mixed up. And then finally, we're just using that save command. And so now if I play this script, you can see here it's loading, it's processing, and that's it, it's done. And so very quickly we can process large amounts of data using these types of scripts to do things automatically for us. Okay, so let's actually do something with these images that we've created. So now we can set our input path to the folder that we made these MIPs in, and we can actually very quickly look to see that they're there. So if I go into our shared folder and go into our cells, you can see we have a folder here, MIPS, and inside we have an image for every single one of these time points of this cell mitosis dataset. And now let's do something. So we want to make sure that we import these images. These images might have different names. We want to make sure that they're in order. So the first step here is we're saying we're just sorting through and getting a list of the files that end with TIFF, and then we're sorting them based on their naming. So we're using alphanumeric ordering. It'll be image 1, image 2, and so on. And then what we want to do is look at all these images together as one. So we're going to create a figure that looks at all these images. And so here we can say, okay, well, the number of images. So whenever we have a list of things, we can say, what's the length of that? And that will tell us, okay, that there's 10 images or 5 images or however many images there are. And then we can determine the layout that these images should have. And then we can create a figure based on this. So I don't want to spend a lot of time here. I just want to show you that just with a few extra lines, we can take that folder and then import our images and then create a figure. And so now we can see here we're actually looking at individual images and the name of the image above that. So we can very quickly look at what's happening in a folder using Python. Okay. So this is really just to give you a starting point so that you can understand, okay, images are worked in Python as arrays, and that we can then look at those arrays just as if they're images, and we can look at their shape and start to do things in an automatic fashion. And so with that, I think now we can move on to the second Google Notebook, where we start to explore how to restore images using care. Okay, so for our second Colab notebook, we're looking at how to use care, and we'll be using the example code that they shared with this analysis toolkit. And so everything I'm showing you here has come straight from the csbdeep website. So whenever people develop good Python tools, they often have very good documentation and provide some example code of how to use it. And I think this is something that I'm hoping we can work on removing the barrier to using these tools, because once you understand some basic Python, you can work with these example codes and tweak them a little bit to your project and really start to use state-of-the-art tools as soon as they get released, if they're released by people who care about them being disseminated. And that's definitely the case for the csbdeep care package. And so for us, to start with in this Colab notebook, we have everything we need in terms of NumPy and TIFF file. All of that's already installed with Google Colab. But what we don't have is the csbdeep package. So the very first cell here, you'll need to run so that we can install csbdeep. So csbdeep is the toolkit that includes the image restoration care package. And so it just downloads that to the Colab and then it installs it. And then this will just take a few minutes, and then we'll be able to start looking at the next cell. Okay, and so in here, these lines are really straight from the care website itself. They're really just importing packages that we need. There's some things here that help with formatting to make sure that the images that we're looking at in line as we're generating them look correct. We're importing TIFF file in read, so this is already familiar to us. And then we're importing specific things from csbdeep. So these are just packages to actually download the example data and then to create patches on that data. So, okay, let's run this line here. This will be very quick. We need to connect our Google Drive if we haven't already. So please do that. Okay. And then what we'll do is download the example data that they provide. I think it's good just to work with this first so we can kind of get a feel for what's actually happening. And so this will be a fairly quick download. And then let's start looking at the next cell. Okay, so in the next cell here, we're creating some variables. So these variables actually match onto images. So Y will be the ground truth data for image 10. And X will be training low signal to noise data for image 10. So these are paired images like we talked about before. And then we're just using matplotlib, just like I showed you in the first Colab. They're using it here to create the figure. So all of this is things we're familiar with now. So we already understand exactly what's happening here. And so they're actually also printing the shape, just like I showed you. And so here it's loading those images in. We can see the color map, just like we were looking at before. We can see the two images, one that's low signal to noise, one that's high. And this is just a really good way of visually exploring your data before you start running the script. And I would encourage you to do this. You can do it in ImageJ or you can do it in Python like this. You want to make sure that the image pairs that you're training the network with really are matched one for one. If this image was matched up with an image that was a different set of cells, then the network will perform very poorly once we actually start to try and use it. Okay, so now we need to create these patches. So we have our folder with the data. The model we're working with here, we want to train it to work with patches of a size of 128 by 128 pixels. So essentially here we're saying, okay, this is our raw data. This is a patch size. This patch filter here, you can learn about it in the documentation, but basically if you have an image that maybe it could be a neuron, but there could be a lot of black space in that image as well. We don't want to create a whole lot of patches that are just empty tissue where there's no signal. And so this patch filter here will say, okay, this image has nothing in it, let's ignore this set of patches. And so you end up with a training set that just contains patches with useful information, and that can be a more efficient way of training the network. And so we can also say how many patches we want from every image in the folder. So maybe we just want to have 2. Maybe we can actually take, if they're big images, we should take more than 2. And so this is just really explaining the variable or the arguments here that we need to get this to work. And what we'll get coming out of this will be a set of patches for X and a set of patches for Y. So our ground truth and our low signal to noise. And so if we run this, sorry, it's bouncing around a bit here. So if we run this cell here, it's just telling us, so it's basically creating the patches for us now. Okay, and so now it's done processing images. And so from our roughly 2,450 images, we have twice that many in terms of patches. So we have almost 5,000 patches for training this model. And so we can come down here and just look at those variables. This is all just kind of, this assert is just making sure that the number of X patches matches the number of Y patches. These are just included to make sure everything is going according to plan, because we're not necessarily checking everything manually. These kind of automatic checks are very important. Okay, and so again, I think this is also what I like about the example code they give. There's cells that really just let you inspect the data. And these are very simple, but very useful, so you can make sure, okay, patch 3 here, so 0 to 2, so this is patch 3. This is what the low signal to noise one looks like. This is the high signal to noise. So we can be assured that, yes, these images are matched well. We're going to be able to train the model successfully. Okay, so now we need to set up the model architecture. So these are, and this is where you may get bogged down with, there's a lot of text here. It could be confronting. This is, again, just keep in mind, this is code that's provided by them. You don't need to know this off by heart to be able to use the tool. All we're doing here is, just like the example I gave before, like where we're importing paints or whatever, if we're creating an artwork, here we're just importing tools that are necessary to create the model. So we can, if you spend some time, you can see they're actually, they make sense. This is loading the training data. This is reading the images. This is so we can see the history of the model as it trains. Okay, and so now we want to actually to set this up. So we've got our training patches here. This is an important step where when we're training models, we need to retain some data to sort of validate how well the model is working. So if we just train on everything we have, we've got nothing left to sort of confirm how well the model is working. If we show it an image that's already seen before, it's probably going to give a good result. What we want to do is save some images that the model doesn't see and then use those to check how well it's working. So we can save some pairs of low signal to noise and ground truth images, and then after we've trained the model, we use these validation images to say, okay, when I give you this low signal to noise image, what do you give me? And if we see the result is good, then we know, okay, even when the model sees an image it hasn't seen before, it can perform well. And so most of the code that you work with for doing deep learning training will have an automatic step in here where it splits off a validation data set. So we don't have to go and manually create this validation data set. I mean, you can have a separate set off to the side yourself if you want to, but we can just give it all our images, create the patches, and then just say, look, I only want 5% of these images to be set aside for validation. Maybe we want to be really cautious. Maybe let's use 10%. So here we can just change the validation split to be 10 instead of 5. Okay, and so when we run this now, we'll get a new set here. So from our 4,400 images, we have 491 that are set aside for validation. So these 4,423 are for training, 491 for validation. Okay. And then here we're just looking, these are the validation images. This is what they look like. It's good to look just in case the set you've put aside for validation are very different to the training set. That would be good. So it's good to just check, okay, look, the validation images look pretty similar to the general pool of images. So that's a good check here. Okay. And so now we want to, this long line here is all about setting up the architecture, or basically setting up the model for training. And so what we can do is modify this. So the batch size, and this is some things to understand, I guess, when training models. We can break the data sets up into batches, so it'll process one batch at a time. And this can be useful to, instead of just processing one image at a time, you can process multiple. This is like more efficient, but it also helps in how the model is learning. And the other sort of term to understand is epoch. So epoch is the number of times we process a whole set of data. So when we say here 40 epochs, it means it's going to process all of those 4,000 images 40 times before it finishes. And what I've just done here, just so we can get a result within the time frame of this workshop, is I've changed the number of epochs to 3. Because if we do 40, then this could take a couple of hours. But if we do 3, it's going to be relatively quick. And because these models work very well for 2D data, even within 3 epochs, we'll actually start to see a decent result. So I'm just going to get this started. And then while it's running, this is really just the configuration. So it basically loads in the default configuration, and then what I'm saying is here for config train epochs, set that to 3. So basically when we run this first line, we get all of this. These are all the sort of default settings for running the care algorithm. And then I've just come in and modified the train epochs to 3, so it's a little bit faster. But if we wanted to, we could change the number of steps per epoch. We could change the learning rate. These are things that you can fine tune if the model's not working well, but the way this is set up currently, I know that it's going to work very well. So all we needed to change was the number of epochs. Okay, so then let's just make the model based off this configuration. This is just giving you a summary of the architecture. So these are the, this may look a little bit confusing, but it's actually very similar to the information I showed you in the presentation. So here these are our convolutional layers. These are our max pooling layers. So this is where we sort of do the convolutional filtering, then we shrink the image down. So it's really just taking you through the encoding, and then here we have the decoding, so the upsampling. So this is just showing you, this is how many parameters there are in each of these layers. So there's many different variables that have been updated. This is the size of the model. And then this is the most important part. This is where we start to train the model. So when I run this cell, you may see a little bit of errors and things here or warnings, but it will actually run successfully. So if we scroll down a little bit, you can now see this is it working through the first epoch. And so whenever we're training a model like this, we're hoping to see the loss start to get closer and closer to zero. And so we need to have some metrics to align or basically optimize the model. So we have our low signal to noise images and we have our ground truth high signal to noise images. We can visually look at them and say, okay, that looks better than the other, but we need to have some way of algorithmically telling the model that it's doing a good or a bad job. And so the way that the metric that's being used here is mean squared error. So this is a way of comparing two images. So when the two images are very similar, the mean squared error will be very low. If they're very different, the mean squared error will be high. And so this error, or this metric that we're using here, is basically the model is trying to make these images as similar as possible. And so it's learning the best way to update, the best sort of parameters in the network to have to achieve that. And so as this epoch goes through, you'll see the losses going down and the mean squared error will also drop. And when it finishes now, it will process the validation data and we'll get some information on how the model is working on the validation data. And so in a second, you'll see this update. Okay, and so it's updated, and if we look across to the right here, you can see these are the results on the validation data. So the validation loss is this, the mean squared error for the validation is this, and as we progress through the epochs, we'll see, or we should see, that the validation loss and validation mean squared error will also decrease. And the reason these are useful is if we just report on what's happening with the training data, the model can get better and better and better at dealing with the training data. But what we might, and what we'll often find is it will reach a point where it can improve on the training data, but it stops improving on the validation data. And this is where it's starting to get more focused on optimizing for the training data, but it's becoming less useful at dealing with cases that are outside the training data. And so if we sort of kept training, we can overtrain the model, and it becomes very good at working with that training set, but becomes very poor or poorer at working with novel images that it may see. And so the reason we keep track of both of these is we can find a point where both the training data and the validation data have improved and then they've plateaued. And so that's the model that we would keep for processing our data. Okay, so we'll just let this run for a few more minutes and then we'll be able to plot the progress and then start to evaluate the model. Okay, so we're finished training the model with our 3 epochs, and if we come across here, we can see that over time the loss has been decreasing. The mean squared error has also been decreasing, and it's the same for the training data, of course, but also for the validation data. There's only 3 epochs, so we definitely haven't trained the model sufficiently yet to see this decrease as much as we would expect, but you can see that every epoch we are seeing an improvement. And what we can do is plot this so that we can see like the course over time. We'll only have these 3 data points, but you can see that the loss here is decreasing and then the validation loss is also decreasing. And so, in my experience, around 60 epochs is a good number to run these. After that, you'll find that the improvement for the validation data will plateau. So yeah, around 50 to 60 epochs, you'll get a good result typically for this type of data that we're working with. All right, and so now comes the validation or the evaluation. So here we can process, so we're creating a figure and let me just explain this. So we're setting up a figure And this here is a new image that we're creating by predicting with our model that we just created. So we're basically saying from our validation data set, pick out 5 images. So this 0 to 5 here. And then for each one, run the prediction. And then we can see here, this is the input patch from the validation set. And then even just after 3, so this is the ground truth, and then this bottom one here will be the predicted result. So you can see that even after just 3 epochs, we're getting a result that, it's almost useful. It's definitely a lot better than the raw noisy input. And so after 60 epochs or so, we actually should be able to really have a better contrast to isolate these nuclei. Okay, and then finally, if we want to export the model, they provide an example here where you can actually export a model. We have to give it a path, but basically we could export our model so that we could use it in ImageJ if we wanted to. Okay. I guess the most important thing, and the thing we're all sort of here to learn, is how to actually use these models on our own data. And so this is just, again, using the code that was provided by csbdeep for doing this. We're importing all the packages that are necessary to predict or infer high signal-to-noise images from low signal-to-noise images. And so, as an example, I've got my ground truth and low signal-to-noise image here that I'm importing. Let's just look at them again. So, very similar to what we did before. We have our model. So we're just importing our model. So this is, now that we've trained a model, it's just a file or a folder that we can carry around with us on different computers or store in the cloud. And when we want to use it, we just use this line of code here. So we basically say my model equals, and then we have our care function here that basically imports the model from this directory. And so you can have different models for restoring neurons, nuclei, or whatever your research entails. And so here I've imported the model, so it's very quick. And then to use it, it's also very simple. It's just one line of code. So we basically say our restored image equals, using the model predict function from the model we just imported on the image that we loaded in. So here X is our low signal to noise image number 10. So if we run this here, you can see half a second of processing time to predict on that image. And then finally, just creating a figure to compare the low signal to noise, the ground truth, and then this is our result. And so here's our resulting image, and then this is just an example here where we're plotting a line through the image and just seeing, that this is the noisy image here, and then the orange line here is our restored image. And so you see we have much better signal to noise in this case. The other thing I wanted to share with you, just like we were doing in the training of the model, we can use metrics to compare our images. Maybe we're training a new model and we want to report in the paper how well the model performs. So we need to give some metrics. And so that could be that mean squared error that we were looking at before. And so here in this case, that's just something that's inside a package called scikit image, which we can talk about more in another workshop. And we're looking at this normalized root mean squared error function that we're importing. And so here, the difference between the low and the high signal to noise, the sort of ground truth image, is 0.6. And then the difference between our predicted and the ground truth image is 0.2. So we can see that by using our prediction model, the images become more similar to our ground truth image. And we can measure that improvement by comparing these numbers here. Okay, that's all I wanted to show you in terms of using care with the example data. Now we can actually learn how to use care for our own data. And so in the next Colab workshop that we look through, and so in the next Google Colab we look at, we'll be using data that we've acquired at OSU to train our own model for restoring neurons. Okay, so in this final example, I'd like you to work through using the knowledge that we have from our first two Colabs to create your own model for restoring neuron data. And so to help you get started with this, I've created some data of high and low signal to noise images, just like the ones I shared with you in the presentation. But I would encourage you that after you finish with this workshop, especially if you're working with data that is noisy, or you're trying to increase the speed of your experiments, or you're working in live tissue or live cells, I would certainly encourage you to create your own training data sets based on the knowledge that you've gained from this workshop. And then you can use this Colab workbook to then train your own data. And what I'll show you at the very end is, now that you understand how to use these Colabs, there's many resources available that people have created Colabs for doing just this type of work. And so I'll show you some great examples of how to access those too. So, okay, let's get started here. So so we need to connect our Google Colab, Google Drive, just as we've done before. So you should be getting pretty familiar with doing this now. And you'll find in the folder that I shared with you a set of paired images of neurons. Okay, so once you've connected the drive, before you start to work with the code from the previous two Colabs to create a model for restoring this data, I also included some additional code here to help you with modifying or augmenting the data that we have. So a lot of times with deep learning, you may have a limited training set, and to expand that training set so that we have more images to train on, and also to help make sure the model is more generalizable, so it's better able to deal with new images, what we can do is augment the data. And what this means is we take one of our images, or patches of those images, and then we apply transformations. And these can be rotations. Just rotating the image gives the model something new to look at, to start to learn how to restore or segment an image. But it's also helpful to add other types of augmentation. So some classic examples are to add shot noise to the image to reflect more noisier conditions when imaging. Gaussian blurring, maybe if the objective is a little bit dirty or the image conditions are a bit poorer, then the model also learns that when it sees a data set that's more blurry, it can still restore to this nice high contrast image. And so the way to do that is to use augmentation code. And so what this code will do is basically create these augmented images for us. And so I've just set these paths. These will be in the shared data that I've shared with you for the workshop. You'll find a low and a high signal to noise folder. And we're just creating these augmented folders as I described. Okay, and so you can see here it's processing them. So just like before, we're using the same Python code that I shared with you before. So we're using F and then the curly brackets to print out the progress and exactly what's happening. So everything in here, if you spend some time looking at it, you would recognize very similar code to what we went through in the previous Colab. So even though it's a lot of different lines here, you would find that if you spent the time, you would actually understand what's happening quite clearly. And so here we're just printing out line by line what is happening. Okay, so yeah, please go through and try to use what we have from the example code, and then match it up with the images that we have for this example. And then what I'll do is I'll go through with this Colab and show you a working solution. Okay, so let's use the example data that I shared with you. Let's go through now and just look at the solution for how we can do this ourselves. Okay, so hopefully now you've had a go using the example data that I shared with you and building on the previous Colab notebook to process that new data. And what we can do now is just go through together using that example data, especially building off the augmented data we made in the previous cell, and see our own model working on our own data. So, okay, here we just have the packages for the operating system to basically create folders and read in files. We have NumPy to work with the arrays. We have random here just so we select random images, and matplotlib so that we're plotting the images in the notebook here. We have our directory set up, and basically we're working through the files that are inside the augmented directory and randomly selecting one so that we can then plot it. And just like in the previous code, we're going to look at a set of images just to make sure that the ground truth and the low signal to noise images are matching up correctly. So let's just run this and then have a look at the output images. Okay, and so yeah, these are actually quite large images, but you can see here, these are our low signal to noise images, and in fact these actually have not just normal noise, they actually have some fixed pattern noise that's coming from the sensor that we're using to do this imaging. But we can see very low signal to noise images here and then our high signal to noise images. And so we've got a diverse, whenever I'm training these models, I try to have a diverse range of images. So I have here some with cell bodies and dense dendrites. I have others where we're just looking at kind of projections and more sort of tissue background. And so it's good to take patches from different areas, and some have very low signal, some have a little bit higher signal. So yeah, I think that the diversity is important in getting a model that works well in a variety of conditions. All right, so now that we've loaded those in, let's scroll down, and okay, just like we did before, we need to install csbdeep. So when you're working with a new Colab, you're starting fresh, and so you'll have to install any other packages that aren't already installed. Okay, so now that we've installed csbdeep, this cell should be looking very familiar. This is the initial cell that we used before to import the example data, but now we've modified it to work with our data. So in the base directory here, you should have the sort of base folder where we have our image data stored, and then for the source directories and the target directories, this can be our augmented subfolder, so where we put the augmented data from the previous cell. And so we can run this now, and it's worked, it's found that data. And so if we want to sort of look at two of these images, we can just, we're looking in the base directory here, we're looking at augmented, low and high. And so if we look here, these images should be matched. So you can see, yes, like these two features here are the same in the low as they are in the high. Okay, great. So the next thing we want to look at now is creating the patches. And so, the images we're working on, if we check the shape, we'd find that, we have it here, they're 1500 by 1500. So when we're creating these patches, we could actually take many more patches than the two we're doing right now. You could make that 10 patches or even more. And so I'm just leaving it at 2 right now to keep the data size small. But when we run this code here, you can see that we have 33 images. So we started with 10, we did some transformations or augmentations, and now we have more. And once we patch those images to create the training patches, we actually have 66 patches. Just as an example, if I do change this to 4, then, and let me just stop this and rerun it, then actually we'll have 132 patches. So you can see very quickly how we could expand the training set to have more patches for training. And then we're just letting the care package take care of making these patches for us. So it's almost complete here, and then we'll move on to the next code. Then we'll move on to the next cell. Okay, so we've saved our training data into this NumPy file inside our base folder. We can double check to make sure the shapes match. Again, they should, so we've got 132 patches in both the training and the ground truth. We can use this cell here to look at those patches just to make sure they match up one for one. So I think this is really important to include this code in any subsequent models that you train, just to make sure like you are really training patch for patch. And you can see here we've got a very low signal to noise image and then a higher signal to noise image. This one, the noise is so low that it's so high that it's very hard to make out this structure, but if we actually zoom in, we can see that feature there. And so yeah, we have some good diversity here. All right, so now we can just set up everything we need for training the model. And so here we're just importing the modules for training. We can run this cell here, and here I'm actually using 20% for validation data. I mean, it depends how many images you have, but if we have a small data set, maybe it's good to have a higher validation split, just so we have more than just a few images to use for validation. Let's now plot those to have a look. And you can see the data set we have here, maybe it's not, real world data is always a little bit more challenging, right? So the example data we had before was very nice, images of nuclei, and then a fixed amount of noise on those. Our training set here, we have quite a bit of diversity. We have some images where, okay, I can see this is low signal to noise, this is high signal to noise. Here it looks like there's almost not enough image information to be able to restore those features. If I was having unsatisfactory results, I might have to go back and actually look at my training data and remove the images that are like this. Especially for 2D, this probably isn't sufficient for restoring this information. In 3D, you can have very little information, but you'd be surprised that as it works across the 3D volume, it can actually restore a lot of structure. So again, this is something that you'll have to explore empirically, but I would say that this is almost not enough information in this patch. Okay, so we can again set up our model. So this should be looking familiar. Again, we're just modifying it so that we're doing 3 epochs. We can print out those settings here, and then we can set up our model. If we wanted to, we could call this something different. So we can, rather than creating a new model that will override our previous model, we could say this is our neuron 2D model, and that way we know what it's for. I mean, when I set these up, I really like to give a lot of information, so I can say this is neuron 20x. I might say 0.3, or let's just say 300 nanometer pixel size, so that I know exactly what sort of image I'm working with. And what the model should see, and then I might want to put in here that it's care 2D. So just to differentiate this from a model that's been trained for 3D data, and then it's always good to put the date in here as well. So we can put a specific date for when this model was trained, so that you can actually differentiate it from other models you might train on this data. Okay, so we can set that up, and then we can start training. So now it's starting to train using our augmented data. We should see it start to come through here now, and then this is it working through the images, and yeah, we'll just give this a few minutes, and then we can look together at the results. Okay, so now we've finished training, and again we can see if we come across here, hopefully, yeah, so this is definitely a more difficult one. So the loss went down, it went up a little bit. Again, it's still lower than it was. But yeah, so this really isn't enough training to get a good result. So we may not have a good result to look at just here, but if we train it for longer, we will get a better result. And so we can just look at the validation loss here. So the training loss is going down. Validation is still a little bit not really improving very much, but with images like this, it's a little bit more challenging. You'll just need to train the model for longer. And so after we finish for today, I encourage you to run by updating this line of code up here back to say 60 epochs and let it run all the way through, and you'll find that you'll get a much better result. But let's just have a look at what our results look like visually for the time being. So we can, again, just using, this is modified from the code that csbdeep shared. So here we have our input low signal to noise images, and so we're just working on some of our validation data. This is the ground truth, matching ground truth, and then here we have our corresponding restored images. So you can see we are getting some restoration, but it's having some trouble. And this could be also because we added in some augmentation, we've got blurring, we've got additional noise, so it's a more difficult data set to work with. But my experience is like giving the model enough epochs with care, you'll be able to train a good high quality model. Okay, and then just like before, we can compare using the mean squared error. I'm not expecting a very good improvement here just with the 3 epochs, but once you've trained this with some more epochs, then definitely try and run this and see for yourself what the improvement is. Okay, and so with that, I'd like to wrap things up, and please feel free to reach out with any questions about using this in your work. I think this is Care is a fantastic package to get started with for image restoration. And then in the next workshop, we'll focus on segmentation tools. So we're looking at how to create masks over cells and in both dense cells and sparse cells. And actually, one last thing I wanted to show you, now that we've gone through using Python, so we have a very basic understanding of what to use to work with arrays and images. We know how to use this care package. I wanted to share with you a website that has a great resource for pre-built Colabs using deep learning tools, which I think is a fantastic resource to use now that you have this initial skill set. And so I'll just share that with you. If we go to zero cost DL for Mike, so Enrique's lab is a fantastic lab working in this space to create tools, but also make tools accessible to a broader audience. And on their GitHub page here, if you scroll down, there's a link. So this is the paper. And if we just go to the wiki page here, this is where we can find links to all the Colabs that they've been building. And so these, will build on what the tools, when tools are published, they may share some code, just like the code that I shared with you today for care. And what this project has been doing is taking that code, just like we did today, and making it into a more usable Colab, so that you can come along without any coding experience and perform the same kind of tasks in Colab. And I think, especially since we've gone through some of the more finer details of what's happening under the hood, you can have enough skill to really come in here and understand what's happening using these models. And so here, these are the different networks. So these are segmentation networks. We may look at some of these in the next workshop. And if we scroll down here to denoising, you can find there's a care 2D. So this will be very similar to what we did today in the workshop. There's one for 3D. There's some other denoising, RCAM, which is the method that I talked about during the presentation. And then there's other models in here or other networks in here for training other models. So I would encourage you to look at this resource to learn more, and definitely start thinking about applying this to your own research. Okay, thank you for your attention.